Thank you very much, Felix. And I'm delighted to be here today to talk to this group, the group that is going to actually solve some of the tragic problems that I'll put before you today. But in terms of the solution of those problems, if I could maybe just seed some pathways and give you some sense of how we're thinking about these problems at the Gates Foundation, some of the strategies that we've adopted, how we've tried to apply some technologies and analytics to some really tragic human conditions. So with that, I'm going to talk here, not for very long, probably about 20 to 30 minutes, so we should have ample time for Q&A afterwards. One of the core principles of the Gates Foundation is that we believe that inequity in health is really an injustice, and that everybody deserves to have a healthy and productive life. And with that tenet, you really have to look at introductions of innovations with a certain sense of optimism. You have to be an optimist, and we say an impatient optimist, if you want to impact and really move things in these lines. But when you're introducing interventions or products into a complex world, it's not just good enough to be an optimist and project to the best world. In that projection into the future, you need to consider all of the downsides, the negative, the obstacles that may happen, so that you can anticipate them. And one of the themes I think I would like you to take away from this talk is how we are now trying to, and how we have the tools actually, to do a much better job of projecting into that future state and understanding what the upsides and the downsides might be of where the world might be in 10 and 15 years. Because that is the world that you're going to be introducing the innovations of today really into. Just to give you a little bit of a sense of what the Gates Foundation is about, we have the tremendous privilege of being able to allocate about $6 billion a year to a variety of philanthropic causes. In the health space, we invest a little bit of over $600 million in R&D per se. My group then allocates about a similar amount to a variety of delivery of those kinds of interventions, whether it be the delivery of how you deliver bed nets into Africa. So that's about that same amount. Now you might say, well, that's a big gap then between, say, 1.2 billion and 6 billion. But we have a number of other areas that we move in. We work on education, which thus far has largely been focused on education in the education system in the US, but we are looking at that more broadly as well. And then there are a variety of ancillary areas. I wouldn't call them ancillary, actually, because they are somewhat fundamental. So take agriculture, for instance. Animal and livestock husbandry. Critical in terms of health from a nutritional perspective. Financial services for the poor. Water, sanitation, hygiene. Family planning and delivery of contraceptive options to women around the world. So there are a variety of other areas, and as much as I'm going to focus on some of the hardcore health areas here, there is an entire spectrum of entities and of issues that need to be addressed before you can tackle what might seem like a disease-based issue, HIV care, or community-based healthcare for delivering HIV treatment, for instance. So I'm not going to talk about that wider set of issues. It may well come up in the Q&A. I'm going to focus here on the healthcare aspects. So that gives you roughly the dimensions of where we're at right now. If we take the way that the world has moved, and this is data from the latest version of the Global Burden of Disease, which is put out by our very good partner, Institute of Health Metrics and Evaluation, that now annually puts out this analysis of what the burden of disease is in the world, what the state is, and what the major risk factors are that drive those various diseases. And it's a changing game over here. There's no denying that. Even in lower middle income countries, I just picked out this one cluster of countries which are now designated by this term SDI, Social Demographic Index, which is really fertility, GDP, and educational attainment. So it's these three factors. We've now found that just judging countries on the basis of GDP doesn't capture an amazing example like Sri Lanka, which on GDP terms it's outperforming on healthcare delivery and healthcare outcomes. So SDI is what we use now to categorize countries in terms of their health outcomes. But if you take the lower middle SDI countries, then what you can see is that there is a drift over here, pretty strong, towards the non-communicable diseases. You see the rise of cardiovascular disease. Nevertheless, even in this group of countries, you can see that diarrhea, pneumonia, still sits at the very top. And if I were to drop you down, which I'm not going to do, to the lowest group of SDI countries, then the focus on the communicable, the infectious diseases, is still even stronger. So yes, there is this very strong global trend towards the burden of disease falling on non-communicable diseases, which I would include the mental health issues. But in the poorest of countries, that is not yet the trend. There is a major unsolved problem. If we look at this projection, and this is going to be the archetype of how we're going to look at projecting into the future, it is a graphic which comes from the Goalkeepers event that we sponsored at the United Nations General Assembly last year. And we will have a repeat of that every year until we make progress, right through 2030. So this is intended to focus on 18 sub-issues of the SDGs, the many, many SDGs, but to focus the world's attention on just a subset of those, and then to present with data where we are in terms of the SDGs, the 2030 goals. And if you take this overall deaths of children under 5, we see this pretty amazing result since 1990 of the better than halving of those childhood deaths. We think that certain initiatives like Gavi have been instrumental in allowing that progression to occur. You do see now in the projection forward, and this is what I'm talking about in terms of, it's not good just to project to the median estimate of what we think is going to happen in 2030, but you need to see what is the worst case if factors don't go our way, and that is the red over there, where we would still be at 3.3 million, versus the upside, the good outcome. Everything goes, the stars align, and then we're at still 2 million too many. But think about the differential between the good outcome and the bad outcome. And that is very much within very accessible, very achievable levers, factors that we can impact, that we arrive at that good outcome and don't sit at the bad outcome. So let me say right up front here that poverty, extreme poverty, is possibly the major factor in driving almost everything that I talk about. The 3 major diseases I talk about are not extant here in Cambridge and in the UK and in the US to the degree that they were HIV. That is a tremendous success story in terms of how the epidemic has been contained and reduced. So the alleviation of poverty would fix all these problems. We see that tremendous progress has been made. We see also this worrying projection that that progress is flatlining through 2030. So at the back of our minds, as much as I'll talk about other things, this we should bear in mind, because progress here is what could really across the board drive improvements. But I think as soon as you say that, there are many complex factors that drive the poverty equation. None the least countries and populations, communities that may be suddenly plunged into poverty because of conflict situations. And the unfortunate circumstances, we don't see those conflict situations decreasing. So this may, at least in that context of conflict situations, be with us for a long time. On the other hand, what we are particularly interested in is not waiting the 50 years, the 60 years, for this general movement of development to solve these problems. The questions that we have are largely around what can we do now on the intervention, the innovation side, to actually accelerate things beyond waiting all that time? How can we have results in 5 years and 10 years on many fronts? The one question that I get recurrently is, probably the most common question I get when I speak to various groups, is how on earth do you decide what you invest in? And quite often it comes from somebody who has a proposal that we may have rejected. And I can understand that because we get many fantastic proposals which may just not be in the strategic areas that we're working in. And probably there are more proposals out there that are worthy than the ones that we've been able to fund. But it is a sum-zero game, and unfortunately in somewhat a diminishing funding game in the development aid area and in funding of global health R&D, although we see some light at the end of this tunnel. So what I did over here was, because it can take a little while to go through this, and I'll go through how we are looking at this problem of allocation of resource and trying to make it a little bit systematic. I did include in your, at least the, Felix, I think you included in your package a little handout which gives a brief description, maybe a little bit more detailed description of how it is that we are allocating these resources. So if you want to dip into that a little bit more deeply, you probably will find that in your package. But essentially, so these bubbles over here, the size of them represent DALYs, so disability adjusted life years. I know it's not a uniformly accepted way of measuring outcomes, but it is one simple summary measure that we've decided to adopt. It's fairly widely used. You can break it down and you can have many alternative suggestions, but it is one which is relatively simple. It is the cost in lives lost plus effective lives lost from disability. And then we can get into questions of how you assess what a disability is from a particular affliction. So the bubble size indicates the DALY size of the burden of that disease. And some of these are large. Some of these larger red bubbles are of the order of 100 million DALYs. So these are large entities in terms of that amount of disease burden. And then there are two axes. There's a third axis, which I'll mention as well, but the two axes depicted over here, one is just this probability of success. And that's a somewhat subjective assessment of overall, in terms of the technical difficulty of doing this, where it stands, what is the likelihood. It's based on a database of similar types of interventions, and you can always adjust that and move that a little bit by 5–10%. But roughly, something very new that's never been tried before is going to be well over towards the 0 to 20%. And something that's well known, a rotavirus vaccine, of which there exist now at least 4. The next one, has got a much higher probability that you'd be able to produce that thing. The other thing being this cost per adjusted life year, DALY, averted. So how much does it cost you not to lose a DALY effectively? And if you can look at the benchmark over here, $100 per DALY averted is pretty cost effective. That's roughly where we are with bed nets to prevent malaria. That you can see is where we are with a little bit better than that treatment for HIV prevention. preventing other people from getting infections and having problems with opportunistic infections, going into hospital. That's where the typhoid vaccine is. And the good news is over there that we have a new, very effective typhoid vaccine, which encourages us to think about, can we actually eliminate typhoid? It was pre-qualified last year through WHO. It's a conjugate vaccine, which should last a long time, can be given to kids. So we'll see some changes on that typhoid front over there. I put down there are two examples I'll talk about over here, but the one in terms of how we use this, we worked with a group in Australia, Scott O'Neill's group down there, on developing this dengue fever vector inhibitor. You could call it a dengue fever gene drive, but it's more a natural, it's not a genetically modified mosquito, but it's a mosquito, the Aedes mosquito, that you've introduced a bacterium into, which has modified the mosquito so it's less able to transmit dengue fever. And that is now in some very exciting experiments, because it turned out that dengue fever is in the class of flaviviruses, which includes Zika, yellow fever, chikungunya. And as we were busy doing some of the initial studies for this dengue fever blocking construct, we were doing those in Indonesia and Vietnam, we suddenly had the crisis with Zika. And Scott quickly went into the lab and looked at the transmission blocking ability against Zika. Actually turned out it was better than against dengue. So we had the surge of interest, particularly in South America, and there's a sizable deployment going on now in Rio de Janeiro and in Medellin in Colombia. And we'll get that data within the next 6 months, whether this is really dampening down. Of course, we've had the peak, and we're now on the downside of Zika, but in Medellin, for instance, both chikungunya, dengue, and Zika now are endemic. So we have a very powerful study over there, and it's more than a study, it's an actual deployment in that region, and we'll see whether this dengue fever gene drive construct actually worked in that context. But then we want to look at that, well, yes, it could work, but is it really a cost-effective thing to do? Because there are other things you could do with that same amount of money. And you see in the best of cases over there, where we target it into the areas where dengue is prevalent and where you might expect outbreaks. And we include, which you could imagine reasonably, the medical cost savings, the few percent of people who are actually admitted to hospital for really severe dengue fever, and some of those even leading to, unfortunately, death from hemorrhagic dengue fever. So if you include the cost savings from that, then you come up with something an overall intervention which would be cost saving. Much like voluntary medical male circumcision to prevent HIV transmission is virtually like a vaccine. It's 60% effective. It saves hugely in terms of infections averted. So it would be up in that territory. And then we get various options where, let's say, well, these cost savings are dodgy. Are they really realized? We can see that it comes down, still cost effective. Then if you did a worldwide rollout, a sort of an unmapped, untargeted rollout, you start to get into not a particularly cost-effective deployment. So this gives you an idea of what you should be thinking about realistically in terms of, do you want to invest in that option? And we'll have to make that decision before the end of this year when the data comes out. I always put up this example, which in fact should come off over here, but you might have noticed this little dot that's sitting at the bottom over there that says Hookworm Vaccine. Now that's an exciting concept for a lot of folks. Imagine hookworm, which causes quite a lot of disability in terms of maybe the major cause of anemia across the developing world. And all the consequences around school failure, the consequences around predisposition to childbirth problems. Imagine you could have a vaccine that would fix that problem. It sounds like a very exciting prospect. Well, there are a couple of issues and I found that as well. So this vaccine program was well underway for a couple of years when I came to the foundation. And then we started to unravel what exactly does this mean. First of all, there's never been a vaccine produced against a worm. essentially. So you have to think, what is the likelihood that this vaccine is going to be 80% effective single-dose vaccine? Very unlikely. Maybe there is something, but it's going to be more modestly effective. Multiple doses probably need to be given of such a vaccine. And then the one other thing which I haven't mentioned, all of these bubbles are, these are bubbles which are relative to the current standard of care. So these are not absolute bubbles that you see over here. They're depicted as relative to the standard of care. And the standard of care for hookworm is the mass drug administration, which goes out every year, has reached billions of people, actually. And there's a drug, albendazole, which is tremendously effective. And there's this deworming of huge populations that goes on every year. If you think about that very effective existing option, then unfortunately, this magical hookworm vaccine looks very small in terms of the incremental DALYs that you would save from that. And if you see at $10,000 per DALY averted, it's massively not cost effective. So very sadly, because there was a big community that was engaged around this, we said we're going to have to terminate the hookworm vaccine program because we can't justify investments in that when these other big entities are options for us to invest in. I do want to go through these three entities over here and just give you the projection of where we are and maybe just touch on one or two examples, mostly just one example of the kinds of interventions that we are working on. So with malaria, this was the map where malaria was endemic in 1990, and we see in 2017 a significant improvement. You maybe think, well, not as much as we might have expected over those 27 years, but there were areas that were really tough that great progress has been made. Take Mesoamerica, for instance. And one aside, I would say, in a direction which is a little bit of a tangent here, but I think is highly relevant to the debates of today, which is that if we think about climate change, another map from IHME, is we could see not just the reductions, but there's potential now for expansion of the regions that could support the malaria vector. And if I were to put on here, now you can see that that involves the US, involves pushing into very close to Europe. If I were to put in here dengue and the Aedes vector, then the extension would definitely be around southern Europe. Aedes has even been detected, I think it's an idiosyncrasy here in the UK, and much of the southern US would be susceptible to the Aedes vector. So this is a factor that we have to bear in mind. The global burden of disease for the first time this year will include temperature as a risk factor. So this malaria situation is one that is pretty troubling. And this came out with the World Malaria Report last December. So what we see here is, you see the peak, and roughly this is about the time that the Global Fund was established. And the Global Fund really energized the delivery of known, highly effective interventions like bed nets introduced into countries. And you see this very good decline. Now, these are cases of malaria, so it doesn't reflect deaths from malaria actually have dropped quite dramatically because of treatments, but these are cases. What we have seen now, and people are arguing about the numbers, 50,000 this way, 50,000 that way, is a plateauing of the progress in malaria over the last 18 months to 2 years. And that plateauing has particularly been evident in Africa, in equatorial Africa. If you think about it, there are 2 countries in Africa, Nigeria and Democratic Republic of Congo, which drive 40% of the deaths in Africa, the 450,000 deaths in Africa, driven by those two countries. And if you take 10 countries in Africa, plus one, that's India, you have about 70% of the malaria cases in the world. So this is a relatively focused problem, but it's in difficult circumstances. Now, troubling over here is that there's a lot of upside to be had between now and 2030 if we can re-energize. One of the factors that we know has been responsible for this plateauing has been some failures in the logistics in getting bed nets basically into those two countries. They've been a big part of the flatlining, DRC and Nigeria. So we can get the bed net supply moving again. In the background, there's pyrethroid resistance. So we now have a new generation of nets that are just being introduced this year, either dual insecticide nets or with some novel non-pyrethroid insecticides in them. In their first incarnation, like any new product, unfortunately, they tend to cost a little bit more. But then you get the same situation, which as soon as you get volumes, that cost will come down dramatically. And we need this, both the logistics of bed nets and we need the new nets to go in. And then I'm sure we can get back into the good side. The bad side, that's what it is. There's one unique thing about malaria right now, which is very exciting to me. There are a lot of interventions that people are working on. On the vector side, there's some fantastic new, very achievable innovations. There's one thing called an attractive toxic sugar bait. So mosquitoes, female Anopheline mosquitoes, don't only need a blood feed in order to produce eggs. They need a sugar feed. And they get that usually from flowers, vegetable matter. Well, a little company came up with a product which is essentially a encapsulated sugar bait made from natural products, made from dates, actually. It's in a sort of polymer bag that only a biting insect can actually penetrate. So you protect pollinating bees and the like from this toxin, because in the sugar bait, there's also a novel toxin. So the female Anopheline senses this sugar, and it's a little plastic entity that you put on the side of your house. And it lasts for about 6 months. So that's a very practical innovation. So there's a lot of this innovation going on. New nets, new baits like that. There's some new drug treatments and new approaches to drug treatment that are in play. In fact, there's so many new interventions, I would estimate at least 10 different approaches, that the question comes about, how do you make sense of them? Because every geography has got a different mix of vectors. It's got a different state of the population in terms of the immune status of the population, because the immune state of the population depends very much on what their risk is of having resurgent epidemic. So there are a lot of factors. There's temperature as well, the particular humidity of that particular environment. So many factors to take into account. And we have previously had the approach that everybody else has had, which is that we need to do lots of operations research. So we do lots of studies. We focus particularly on Zambia, but we have centers in Mozambique. There are centers in Kenya, and they do, let's take this center and do a cluster-controlled study, where in some of the clusters we do this package of interventions, that other clusters we do the standard, and then after 5 or 6 years, you get a result out that something may have worked or not worked. Well, if we were to go in that way forward, we'll be, maybe the poverty efforts will have caught up with us, because it's going to take an awfully long time to sort out this mix of interventions. We are at the state now of being able to do in silico simulation of the malaria epidemic that allows us in silico to put the packages of interventions together. And we can do these simulations for the first time at the scale of an entire region, an entire country. And we can put in, from geospatial mapping, the realistic geography of the country We can put in the entire history of the climate with the projection of how that climate will be changing. We can, from sampling serosurveys of the population, put into this agent-based modeling the immune status of the individuals. So every factor that could be relevant, we can now put into these simulations, and we can run them forward with different packages and with different assumptions. So we can see, before we do an actual study, what is really differentiating, what package really should show a big difference. So we don't have to sort through the 35 different packages. We can pick maybe 2 that are the ones that are most promising, put them into the operations research, and short-circuit decades of doing this research in the field. This is one example of that, and I'm going to just show you a quick simulation, and just to locate you to what's happening over here. So we're sitting in Zambia, which has done a pretty good job in terms of reducing the malaria burden. And it's one of the countries that we focused a lot of our operations research on. Around Lusaka, the capital, and the surrounding areas, you probably don't even have to take anti-malarials anymore. But unfortunately, both in the north of the country, but particularly in the south of the country, around Lake Kariba, malaria remains endemic and seems somewhat intractable, because we and others have invested a huge amount of effort in getting our bed nets. We've thought about families who've got 2 bed nets. Let's give them a 3rd bed net and see if that works. So whatever we've done has not succeeded in these highly endemic, high transmission zones. And this is the zone around Lake Kariba in the southern part of Zambia. Now, the intervention that we were hopeful of is called mass test and treat. So what happens in a place where you've got a lot of malaria is that most people don't get sick, and particularly adults become quite immune to infection. So you see infections, you see particularly some infections in kids who are less immune. Young kids, the younger they are, the less immune they are. But in adults, they tend to be walking around. 90% of the population in these areas, if you test them, actually have got malaria parasites. So they are the reservoir for transmission of malaria. So the idea is that if you can clear the vehicle, the reservoir, then, and you can actually keep the vectors away from them, that's bed nets, then you should actually be able to rapidly deplete the transmission potential in an area. And that was the theory. But when we went to the simulations and looked at that, so let's see if this actually works. Oh, it does. Okay. So what we are simulating through over here is about 2 years of mass test and treat to these. Now, the dots are little settlements, typically, and the color of the dot is the rapid diagnostic test for malaria, positive. So the red dots are where there are a lot of little communities where people are testing positive for malaria. They are usually coming into clinics. In fact, they're all coming into clinics in this case. And we are testing out this intervention of mass test and treat, and then just watching what happens over 2 seasons over here. And what we saw is that this looked great in the first incarnation. You didn't quite see that over there. But what happened was, in reality, in the actual test, and the simulation actually, if I go back to that, maybe you can see that in the sort of the summary of that. This is the summary of what happened. So you can see the simulation is below and the actual data is above over there. What happened is that after these 2 rounds of the mass test and treat, clear the reservoir, looked like we'd made a lot of progress. But then when we went back to the next season, it pretty much reverted to the state as before. And the simulation actually showed us the same thing. And so what that indicated to us is that this mass test and treat, as attractive theoretically as it is, doesn't actually work if you have pretty high transmission like this. So more to come. Malaria is going to be the area where in silico simulation is going to be widely deployed for global health problems at both a national and a regional scale. Tuberculosis. So this is a very old infection, and some of the data, the old data actually is pretty surprising for a couple of reasons. So let's just take the top graphic over there. So these are deaths. You can quibble about cases and the diagnostic accuracy back in the 1800s and the like. But typically deaths from TB, we think, were pretty well recognized. So you take the deaths from TB per 100,000 population. Let's take New York over there, where you see in 1860 through 1880, you had this peak over there. And then you have a precipitous decline to a much lower level of deaths by 1940. Well, the interesting fact about that is that effective treatments were only introduced in the 40s and 50s, drug treatments for TB. So almost all of that improvement occurred without treatment. And this is the quintessential example of TB is a disease of poverty. And it was improved nutrition, improved housing, basic infection control of removing people who are infected from the general population. So some very basic public health interventions were the cause of this dramatic reduction in TB in New York City. In the 1880s, the late 1800s, very sadly to say, the epidemic in New York was almost identical in its parameters. So we had then, let's say 300 deaths. At that time, about 50% of people with TB actually died. Right now in, for instance, the Western Cape part of South Africa, we have a TB incidence of 600 per 100,000. So we in the state there now that New York City was in say, 1890. We look at the projection over here. Certainly there's been some progress. There's this worrying potential for plateauing over here. And then the other remark I'd make about this is that by 2030, even in the good case, there's still a lot of TB cases around. So this is something that is moving very, very slowly, even in the best of cases. And it is an area which is incredibly underfunded. In most of the global health areas, you take HIV, you take malaria, the Gates Foundation is a very small part of the funding of R&D. The NIH, has got vastly more funding into HIV research. In malaria, there's a huge community of funding of research on malaria, and we are a fairly small part of it. In TB, the sad fact is that we are the major funder of R&D in TB. But there are a couple of simple things that can be done quite quickly. The biggest single change that could happen with TB is better diagnosis. So what we know is that a lot of people who are symptomatic are coming into healthcare systems. This is not finding people who've got TB. These are people who come and they say, I am unwell in some way to the healthcare system, whether it be in South Africa, whether it be in India. Indonesia is the third country now, and China is somewhat, has done a good job in terms of reducing its TB burden. People come in, I am unwell, I've lost weight, I have night sweats, typical complaints of TB, and they don't get tested. And they get sent back. Maybe they got an antibiotic. And then they may or may not come back. In that intervening time, they are transmitting TB. In China, we've estimated that people may be transmitting TB for 2 years before they actually get into a treatment program. So diagnosis, and this seems really achievable, can we not diagnose TB more rapidly and then be able to institute some treatments? And we can, and this is where there's some very interesting experiments. This is the one that I love most of all. It is a young group out of Stanford. Riju Das is the leader of this. He's a biochemist, who got into this idea of crowdsourcing problems, biological problems, to people who are gamers. And he calls this the Turner game. And the way that he's configured this he asked these gamers. Now, there was one time a game around, and maybe it still exists, but I think this is the next generation of that, of asking gamers to solve protein structures. This is about asking gamers to solve RNA structures. Now, because RNA has got more mobility than proteins, that is a more complex problem to actually solve. And if you think about the opportunity out there, I was reading this fact that there are about 200 million of attention given every day to the handheld game called Angry Birds. So what if you've got a fraction of that time to be spent on problems like this? But in any case, what Riju did was he set up this for a TB challenge. And I'll explain to you what it is around. This is a depiction of what happens. This is the RNA molecule, and it's basically looking for 3 components of blood. And when it finds them, it flips its configuration. So what we found is that there are 3 mRNAs that you can detect in blood that are highly predictive of somebody's risk of developing TB. But when they looked at how you have to analyze it, it's a little equation you need to solve. You need to take factor 1, divide it by factor 2, and then multiply, and then I think you add factor 3. So there's a little equation that you need to solve in order to come up with the risk score of these factors in blood. So the idea over here was that your RNA molecule will actually reflect that equation, that its conformation will flip when it detects those 3 factors with the division and the addition condition met. And then you have a fluorescence marker that you can then detect the flipped configuration of the RNA marker. So what Riju's group does is that when people submit solutions to the problem, every week they actually make the RNA molecules and they test the solutions, and then they give the gamers feedback, well, your solution worked or didn't work. So this, we hope, is going to lead to now we knew about the 3 factors, the expression factors that were predictive of TB risk, but we didn't have an easy way to measure them, and we didn't have a cheap way to measure them. So we have to see where this goes. But this is the kind of new approach that you could never have thought about 10 years ago. On HIV, if we look at the now somewhat familiar graphic of projecting into the future, we see a very difficult projection over here. So you see the tremendous progress that has been made, and that has been largely due to PEPFAR and the Global Fund, introducing a lot of money into treatment. So we look to be in good shape, but now you see the projection of plateauing of that improvement, but then you see these upticks over here. And the red downside, the worst case over there, that's a bad alignment of existing factors. Then the even worse outcome, that would be if the US were to reduce their investments in the PEPFAR program as have been proposed, not enacted yet, and hopefully never. But if those were enacted, then we could expect things to be even worse, because that is the major source of treatment for tens of millions of people in Africa who essentially would have to come off treatment. And it's not just a matter of coming off treatment over here. So just to give you a sense of the time bomb that we are sitting on, And this is the demographic time bomb. What you are seeing in Africa over here is an explosion of young people. Now that's the demographic dividend. For HIV, it's the demographic time bomb, because this is the explosion of people in the 15 to 24-year age group who are the risk population for HIV. That population is exploding. So even though, yes, incidence is decreasing of HIV, by 2035, just the demographic trends predict that there will be more individuals who have HIV in Southern and East Africa. And there will be less funding for treatment than there is today if those trends continue. So it astounds me that often when I talk to groups, people say, well, HIV is a problem that we've solved, right? We don't really hear about it that much anymore. It is an exploding epidemic, and it is an uncontrolled epidemic, and there are not a set of tactics that are in place today that are going to prevent that situation in 2035 from occurring. Well, it certainly needs everybody to put their minds together. The particular population that we have the greatest concern about are vulnerable adolescent girls. And how do we protect them absent a vaccine? We have work ongoing with a vaccine. Many of the pharmaceutical companies, J&J is very invested in this, GSK is invested in this, Sanofi is invested in this, so many of the investors at this conference have been working with us towards an HIV vaccine. And I'm delighted that there are, for the first time in quite a long time, 2 major vaccine programs which are in progress right now in Southern and East Africa. It is a tall order to produce an HIV vaccine. So what we typically could expect, as with any novel vaccine, is that these first forays may be modestly effective. One of them is following on a modest result obtained, the so-called RV144 study in Thailand, where it turned out that the vaccine was about 30% effective, requiring repeated boosting. So we might get out of this, I think, realistically, some modestly effective vaccines, but not the kind of vaccine that is really going to completely change this story over here. So what are we going to do absent that we get that vaccine? Well, there are some things that can be done. And one of the ones that I'm most excited about is that as much as the development of drugs for HIV has been quite a fantastic story in terms of coming up with effective compounds, that has progressed. And it's progressed in the direction that we continue to be able to produce, and I can't explain this biologically, but we continue to be able to produce ever more potent and long-acting compounds. So that we have certain compounds which at very small and doses that would pretend that you could put them into some long-acting formulation might be able to protect people for 1 year. So what I'm showing over here is really it's a contraceptive implant, but it's that same idea. So the idea is that you would have one of these highly potent, long-acting formulations in an implant that would last at least 1 year. But the trick to success over here is that it has to be co-formulated with a contraceptive. So this would incentivize people to use it as part of both family planning and HIV prophylaxis. And that gets us into the channel of the young adolescent girls in an achievable way. So you've got a couple of tall orders here, but they're achievable. We can see the pathway. It's going to be difficult. The implant is a different implant. The co-formulation has not been, I would say, brought to a state where I can give you, great confidence that it can be done. From the volumes it can be done. These drugs often don't live well together. So we'll see, but this is pretty hopeful. So this could be the intermediate intervention that prevents us from getting to the worst outcome. I've talked about these 3 diseases, and I've mentioned a number of technologies, and I just wanted to come back to technology itself. How we think about that. So it is actually, we are at the stage where as much we read in the newspapers about these fantastic technologies that seem to vanish, and you can become skeptical about, well, do any of these things actually work at the end of the day? In fact, yes, some of these things do work. So yes, we have drones that are being deployed because they can use spectroscopic analyses of malaria wetlands, breeding sites, and they can identify them more effectively than sending humans around by foot or in vehicles. So yes, drones are going to be a big part of mapping out malarious areas for larvicides that would go into those wetlands. Collecting mosquitoes with robot traps. This is nascent, but the proof of concepts look very compelling, that we have robot traps. They have little trapdoors. They put out a CO2 attractant. Mosquito comes along. Is it the right mosquito? Could be just there are dozens, hundreds of mosquito species. If you just take any one, then you're going to get a very inefficient sampling. But it uses a machine learning algorithm to identify on the fly, is this an anopheline species? If it's an anopheline, it closes the trapdoor and samples it. That robot actually works very well. And that is going to be deployed, I predict, in the next 3 years to sample the mosquito populations in those difficult areas in Africa. And then what we're going to do with those mosquitoes, currently, Sanger, here in Cambridge, is completely sequencing 3,000 mosquitoes a year, everything in the mosquito. So you get the mosquitoes, you get the identification of the mosquito, you get the identification of pyrethroid resistant genes, you get the microbiome of the mosquito, actually, you get the blood of the humans that the mosquito bit. So you get all the viral infections in the human population where you captured the mosquito and other infections in that human population. And interestingly enough, countries which are very reticent about sharing samples have been quite free to sharing their mosquitoes. So maybe this is the under-the-table way of understanding the human populations. So these apparently science fiction approaches are actually here and now. But the caution is that as much as we may be excited and rush to deploy these, we have to really think about the circumstances and what the actual problems are out there. It's interesting to me, we had a meeting just a few days ago with the companies, the pharmaceutical companies, and then we had the funders. We were one of the funders, and we had other government funders and everything. And there was just these two approaches. So on the funder side, people said, well, we have $1 billion, and we're looking for problems that we want to spend the money on. And the company said, that is an amazing way to look at the world. We look at the medical health problems and what it requires to actually solve those problems. We don't think about, we have this pot of money that we are just going to ask people to come in and use in some free form over there. So my heart actually is with the companies and not with this free form type of funding. Because if you want to make progress, you've got to think about the problems. And where we've made mistakes, it's about not thinking about these problems. So take, for instance, TB. We come up with a great set of medications, and then people have to take this huge package of tablets every day. The current TB regimen, I've come to understand, which includes isoniazid and rifampicin, and really is driven by that one drug, rifampicin. That you take that out of the regimen, doesn't work at all. And it's the dose that you get of rifampicin that basically drives that whole regimen. That it is an incredibly unforgiving regimen. It works very badly. You have to take it for 6 months. But not only that, we found in studies now that if a patient misses 1 in 10 doses, they reduce by 20% their chance of being cured. Miss 1 in 10 doses. Who doesn't miss 1 in 10 doses? So what are you going to do about that? Well, there's some things that you can do. Now, it doesn't always have to be complicated. In India, we did have a grand challenge for ideas about how do you do various things. And somebody came up with a very simple idea. They said, well, for malaria, for TB, Let's give people a little card which has got their medication in it like this, and let's give them a date, a little telephone signal at certain way stations along this card, and when they hit on the day of a little telephone, they've got to call this free number to just confirm that they took their medication. And I wouldn't have thought that something like this is going to really make a big difference, but so far all the indications are that it works amazingly well. I don't know what the social dynamic is of why people get their card, and when they get that green thing, we've got a tremendously high rate of people calling in. So sometimes these very simple interventions like this can make a huge difference. And that comes from, I guess, the person who had this idea somehow understood something that we didn't understand about the psychology of people who are taking medications like this. All of the work we do is through a huge network of engaged partners. And almost every time I talk to people, I have to add somebody to this list over there. And then, of course, now what I said is that make it so small that I'll flip through it quickly so somebody won't notice. They can't claim that they weren't on here when they should have been on here. So I'm going to do that exactly. Okay. But I just want to talk to you about two of those partners quickly. And that is from the perspective of the kinds of people that are working in these partner groups. And maybe you can see yourselves in that role, or you can see friends or acquaintances. So Just Biotherapeutics. It's a little company run by somebody called Jim Thomas. He is a manufacturing expert who worked with Amgen for 30 years on production of antibodies. And some of the most important antibodies that have been produced, Jim was involved with. And he decided, during Ebola actually, he started to work with us around some of the antibody solutions for Ebola. And then put together this company, which is funded by us and one or two other partners. It is, outside of the big pharmaceutical companies, the only other place where there is very high quality bioprocess development of antibodies going on. They've jumped in and they've transformed. We've got these antibodies to protect against HIV, the broadly neutralizing antibodies. The ones that came out of research were tool compounds that would never, ever be used in anybody because they didn't have the right development. Just transformed those into something which can be given in a small 1 cc subcutaneous dose, 2 antibodies mixed together, an amazing formulation that makes it now a practical solution. So they've got this fantastic capability. And so you've got the experience that we absolutely need, 30 years of manufacturing experience from that background. But he's got a very young team that is working on algorithmic approaches to speed up the bioprocess development exercise, looking at protein structures and predicting changes that you might make without having to do a lot of experiments. And they've made a lot of progress. So that's a different group over there. The other enterprise is this, what we call the Gates Medical Research Institute, which we've just established in this last year, and it really is going to have its opening in June. It is around translational clinical work in particularly TB, it'll start off with. Malaria vaccines also work on. And in these tough areas where we think we need a new approach to translational work, a much more tightly integrated approach. And it's also going to be an open institute, open research. So we've talked a lot about what is open research. Well, now we have a chance to define what it means to have open research. This is not funded by a pharmaceutical company. It's not got a bias. None of these products are going to, well, they may have some commercial, they will have a commercialization at the end of the day, but the primary focus is not on commercializing products over here. And we have a very strong relationship with the FDA, with the pre-qualification at WHO, and with the country governments and the country regulators. So can we have a completely different mindset around how we do translational work in these public health emergencies over there? Now this is the people who've come into the Institute, a a couple of the senior people have had 10 years in biotech or in pharma as their backgrounds, but they are very early in their careers. And it is a sort of a young, engaged group over there that I'm particularly excited about. I can see many of you potentially in that kind of role. So with that, I will stop my comments and thank you for your attention.
I think we have time for one or two questions.
Good morning, Dr. Mundel. It was an incredible talk, quite inspirational. I'm Professor Melinda. I'm a genomic biotechnologist and I'm a PhD student in international health. So I'm quite excited about your topic. My question is, what do you think is the biggest challenge for implementation of new biotechnological strategies for public health? Because I think it's the biggest implementation is the biggest challenge that we have in the hands now.
In general, for new interventions in public health?
New biotechnological interventions in public health.
You're right. So, at base, what you need is a functioning healthcare system for many of these technologies. And this is what has driven us to focus so much on vaccines. So vaccines, in some ways, you can get around a very dysfunctional healthcare system if they're the kinds of vaccines that you just need one dose of the vaccine, of which those are rare, but we have the expanded pediatric program, the EPI program. So vaccines somehow transcend the lack of a really good functioning healthcare system. And that is a massive challenge. We have become much more invested in that from the foundation perspective. Now, of course, if I give you dimensions of problems, the problems of R&D here, we're talking about $1 billion problems. The problems of healthcare systems are $1 trillion problems. So there's no way that the Gates Foundation or any kind of philanthropic sector or any company is going to solve those problems. It is really a fundamentally problem of sustainability that country governments and the multinational organizations, the World Bank and the like are going to have to are trying to grapple with, but are going to have to do a better job in. We've looked at a country, particularly the primary healthcare systems. If you have an engaged government, like in Ethiopia, the government has engaged quite dramatically in health. There's a lot of progress to be made. We're supporting them in terms of logistics. We believe we can bring some logistics supply experts to that equation. And we've also looked at a scorecard. So how do you know that a public healthcare center is working well or not? So these are just peripheral things, ancillary things. The core funding, it's got to be a combination of the government has got to want it, and often with support from donor governments in many cases. But it is a huge unanswered question.
Could you talk a little bit about the different types of relationships that the Gates Foundation has with the pharma? And specifically your role in the emergence of social businesses?
I'll just tell you a snapshot of that, because we have extensive interactions with the pharma companies. And in terms of getting access to their capacities, this is the fastest path to success for these new interventions. They've got tremendous resources, and in certain areas, they've actually been quite willing to work with us. So we have an entity which we call the CEO Roundtable, where Bill gets together for a dinner and a day with the CEOs of the major pharmaceutical companies. And we talk about how we could do a couple of projects in common. One big project has been around drug donations from those companies for the neglected tropical diseases. That's been a huge donation program, which has made a big difference in those diseases. Another one there is called the TB Drug Accelerator, where 8 companies, including now some academic institutions here in the UK, in the US, the NIH are involved, where everybody is making accessible their drug discovery libraries to look for new TB drugs. This could be a kind of model for doing antimicrobial resistance as well. And that has actually transformed TB drug development. We now have quite a lot of promising compounds that are coming out of that collaboration. We're looking for what the next set of projects are that we could work with the companies on. And they have a certain appetite because in some ways, the role that we've been able to play is as a neutral convener and bridge, both to the regulators and to some of the multilateral organizations like WHO that is, I think it may change now with the new Director General, who's certainly looking at quite a big change agenda, but traditionally they've not had a good working relationship with pharmaceutical companies. So we've been in that role of trying to bridge that divide. Many other interactions, and those companies are critical to our progress.
I think we have to leave it here, I'm afraid, otherwise we're running late.
Sure.
But let's thank Trevor again for a fantastic session. Thank you very much.
Thank you.
Yeah.